Bismillahirrahmanirrahim. In this series of video, we are going to discuss the blistering or bullous diseases of the skin. This video particularly will cover introduction to the histology and classification of the skin and blistering disorders. When we attempt to classify skin disease, we can classify these diseases on the basis of the site of origin or the anatomic structure that is involved. One group is disease of the epidermis, and that include ichthyosis, diarrheal disease, psoriasis, and pemphigus vulgaris. Another group is disease of basement membrane zone, that is the dermal epidermal interface, and this include epidermolysis bullosa, bullous pemphigoid, dermatitis herpetiformis, erythema multiforme, systemic lupus erythematosus, and lichen planus. The inflammatory diseases of superficial and deep vascular bed include urticaria and angioedema, cutaneous necrotizing vasculitis, allergic contact dermatitis, granulomatous dermatitis, sarcoidosis, and granuloma annulare, while scleroderma is the disorder of the dermal connective tissue. The paniculous inflammatory disorders are erythema nodosum and endoretum, while acne vulgaris is also an inflammatory disorder. Then there are infections such as impetigo, superficial fungal infection, deep fungal infections, viral infection, and arthropod infestation, and variety of benign and malignant tumors. Some skin diseases are also affected or also manifestation of the paraneoplastic syndromes. Then there is another way to classify these diseases, and this is done on the basis of the process, the pathologic process or morphological appearance of the lesion. One is the epidermal maturation disorders, and it includes ichthyosis. Then acute inflammatory dermatosis include urticaria, acute eczematous dermatitis, and erythema multiforme, while chronic inflammatory dermatosis include psoriasis, seborrheic dermatitis, and lichen planus. The bullous or blistering diseases of the skin can be divided into two groups: inflammatory blistering disorder, that include pemphigus, bullous pemphigoid, and dermatitis herpetiformis, and non-inflammatory blistering disorders that include epidermolysis bullosa and porphyria. The disorders of the epidermal appendages are acne vulgaris and rosacea. Paniculitis is the group that includes erythema nodosum and endoretum, and other types of paniculitis are also there. Then disorder of pigmentation and melanocyte, freckle, lentigo, melanocytic nevus, dysplastic nevi, and melanoma, and benign and malignant tumor along with adnexal tumors of the epidermis and dermis. Now a brief review of the layers of the skin. Outermost layer is the stratum corneum that continuously desquamates. Below this layer is the stratum lucidum that is thick, and this is pinkish in color. Just below this there is stratum granulosum, and this stratum granulosum has got a granule-like appearance, and it contains purplish cytoplasmic granules, and below this prominent layer of stratum granulosum there is presence of stratum spinosum. Stratum spinosum has got polyhedral cells that have got prominent intracellular bridges, and all these layers rest on a prominent basal layer, and over here this basal layer is showing a brownish pigment that is melanin, deeply brownish pigment that is melanin, and this circle is pointing towards the papilla, uh, upper, that is upper papillary dermis, and here a blood vessel is present, and this blood vessel has got its role in the regulation of the temperature. The basement membrane is the interface between the dermis and epidermis. It has got diverse function and it has got a complex nature. It is responsible for the dermal epidermal adherence, and it is a selective macromolecular filter, and also it is a site for complement and immunoglobulin deposition in certain diseases. And most structure of this basement membrane zone are secreted or produced by the epidermal cells, and the basal lamina is the primary organizational feature of the basement membrane zone, and it is responsible for the epithelial cell polarity and some keratin gene expression. The structures that are included in this basement membrane zone are deep aspect of basal keratinocyte. There is a plasma membrane. Over here you can see that there is the plasma membrane, and this plasma membrane along with tonofilament, and these attaches to the deep face of the hemidesmosome. Okay, then hemidesmosome along with its subdesmosomal dense plate that is marked by this red plate in this picture just beneath this blackish desmosome. Then anchoring filaments. These anchoring filaments extend from the subdesmosomal dense plate across the lamina lucida, and they insert into this greenish lamina densa. Then lamina lucida is composed of adherence protein, while lamina densa is composed of type 4 collagen, dense type 4 collagen. Anchoring fibrils are down there, and these anchoring fibrils are composed of type 7 collagen, and they bind this lamina densa to the papillary dermis. And microfibrils are there also. These are thin reddish microfibrils that are delicate long elastic fibrils. So from outer to inner side, the basement membrane parts are deep aspect of basal keratinocyte, hemidesmosome, anchoring filament, lamina lucida, lamina densa, anchoring fibrils, and microfibrils. So now recall the structures we just discussed. Recall in your mind the structures from above to the down to the dermis. Then this electromicrograph is showing the lamina lucida, lamina densa, and there is a desmosomal attachment plaque that is marked by this arrow, and down there into the papillodermis the anchoring fibrils are also visible. As this basement membrane is a complex structure and it has got a unique morphology, it has got some antigenic sites that are important in the development of the skin disorders, especially the blistering disease of the skin. One antigenic site is for laminin. Laminin is present in lamina lucida and lamina densa, and it binds type 4 collagen. It is a glycoprotein, and it is present in both these layers, the lamina lucida and lamina densa, and it is responsible for binding of type 4 collagen. There is another antigenic site, that is bullous pemphigoid antigen 1 and 2, that is concentrated in the hemidesmosomes and basal keratinocyte. It is important in bullous pemphigoids. Then there are type 4 collagen. This type 4 collagen is concentrated in the lamina densa. This lamina densa is the most superficial component of the complex network of the collagen of the dermis, and it is also important in the dermal epidermal attachment. The type 7 collagen is present on the deep aspect of the basal lamina in the anchoring fibrils, while the anchoring filament 1 and anchoring filament or anchoring fibril 1 and anchoring fibril 2 antigenic sites are present in the anchoring fibrils and lower part of the lamina densa. The pathology of the bullous diseases of the skin is associated with the keratinocyte adhesion molecules. There are desmoglein 1 and desmoglein 3, two important proteins, and one of this desmoglein 1 is more concentrated in the upper layer than the lower layers, while desmoglein 3 is more concentrated in the lower layer up to the stratum spinosum, and it is almost absent in the upper layers. So the antibodies against the desmoglein 1 will result in subcorneal blisters, as happens in pemphigus foliaceus, and the antibodies against both the desmoglein 1 and desmoglein 3 will result in suprabasal blisters, as happens in pemphigus vulgaris. This picture is showing the composition of these desmosomes. This desmoglein that is present in the uh, that is the site of the problem in pemphigus is attached to the intracellular structural protein through plakophilin and desmopalakin, and it is attached with this tonofilament and giving the support to these cells. It is There is another protein also that is responsible for the support, that is desmocolin. While the hemidesmosomes, uh, they have got some specific structural protein. One is the BPEG1, that is bullous pemphigoid antigen 1, and this bullous pemphigoid antigen 1 connects the intermediate fibers with the laminin or and subsequently the anchoring fibril through the bullous pemphigoid antigen 2, that is the pemphigoid that is responsible for the pemphigoid. When there are antibodies against these bullous pemphigoid antigen 2, there is formation of the blisters that are at the level of the lamina lucida of the basement membrane. While when there are antibodies against the anchoring filaments, these anchoring filaments, and these antibodies will result in the disease dermatitis herpetiformis. These are usually IgA antibodies. There is a clear distinction between the two terms, the vesicle and bulla. The vesicle are the morphologic lesions that are characterized by fluid-filled raised lesion less than 5 millimeter across, and the bulla are the fluid-filled lesions, raised lesions that are more than 5 millimeter across. Now, just to revise the classification of the bullous or blistering dermatosis, we can say that these can be divided into inflammatory and non-inflammatory types. The inflammatory are pemphigus, bullous pemphigoid, and dermatitis herpetiformis, while the non-inflammatory one are epidermolysis bullosa and porphyria. The pemphigus is the disease of the epidermis, while bullous pemphigoid, dermatitis herpetiformis, and epidermolysis bullosa are diseases of the basement membrane zone, while the porphyria is due to intracellular accumulation. Then there are some unrelated conditions that also present with the bulla formation. These are herpes virus infection, spongiotic dermatitis, erythema multiforme, and thermal burns. But these are not considered in the blistering dermatosis. These bullous disorders of the skin can be fatal in some instances if untreated. And as we have seen the distribution of antigenic component, these bullous disorder may present with different type of the bulla, and these can be subcorneal blister, suprabasilar blister, or subepidermal blister, depending on the pathological process involvement. In the subcorneal blister, the roof of this blister is formed by stadium corneum, while the rest of the epidermis forms the floor. While in suprabasilar blister, the basal layer is separated from all the above layers of the epidermis, while there is complete separation of the epidermis from the dermis in subepidermal blister.